Nada?
Por causa dessa modalidade aí do...
E não tá aí no próximo slide.
Porque olha como tá ali.
Aí agora foi.
Tá, então foi.
Só que tá assim.
Só que,
Aí não,
rapaz, o que tá...
tem que tirar o modo de apresentador. Agora foi, agora tá. E ali, tá ok? Ah,
We don't have the presenter mode, like you can see the next slide. Is that okay? I'm sorry. Okay. Thank you for yeah. Okay, I need to get my notes.
é da University of Nebraska, Lincoln, e ela está aqui através de um... Então, mais um pesquisador que através desse projeto Capa Springs. Ela fez a graduação na Cornell University, inclusive a nossa parceira do projeto Print. A Bia acabou de tornar de lá, né, fez um sanduíche, e ela fez depois o doutorado na Arizona State. Agora já faz alguns anos. A palestra dela, de hoje, o título é Growing Rocks, How Bioavailability in Aquatic Ecosystems. A palestra vai ser em inglês, mas a gente ficar fazendo perguntas em português, pode traduzir, ou pode fazer em inglês também, se vocês... então vamos deixar a palavra a Jéssica. Jess, thank you very much for being here today. We're just giving a short presentation about you. And so you can go ahead. Thank you.
Thank you. Thank you, Eugenia, for the invitation. And for what I could tell was mostly a very kind introduction. I am sorry that I don't know Portuguese. Otherwise, I would try to speak it. I have attempted this presentation in Spanish, and I am afraid that I would just jump into that anyway. But thank you all for being here. I am talking about some of the work that I started in grad school and have continued on. But before I get started, I wanted to thank people that funded the project, lots of people that helped, and some of the folks that I worked in the field with to conduct a lot of this work. So when I go about my research, I like to think about how I can advance biogeochemical theory and also inform global environmental challenges. And one of the things that you might think about when you think of a global environmental challenge, right, is that we as humans have drastically changed the distribution of certain elements on Earth. One of those is, of course, carbon. We've burned fossil fuels, land use change, increased the amount of greenhouse gases, carbon dioxide, methane, and others. And that has led to profound changes in our climate regime. What you may not realize is that we've had profound impacts on other elements as well. And when we compare those impacts to carbon, proportionally speaking, we have to adjust the scale. So we've increased the amount of bioavailable nitrogen in many ecosystems through the production primarily of fertilizers. And we've also drastically increased the amount of phosphorus, generally through mining activities to make fertilizers to grow food. So they're important to change these, the rates of cycling of these elements, but they have also had pretty dramatic consequences to a lot of ecosystems. This is a picture of Lake Atitlan in Guatemala. And you can see the spirals of a cyanobacterial bloom, a harmful algal bloom in that lake. A lake that is surrounded by mountains is has a very small watershed to lake area, and not a lake that had problems with blooms before the last about 15 or 20 years. This is entirely anthropogenically driven. And of course, Lake Atitlan is not the only lake that has problems with eutrophication or the only aquatic ecosystem. And so one of my questions has been, you know, are there processes that we can understand in streams or elsewhere that can help slow down the movement of these elements? Can we manage our aquatic ecosystems to retain nutrients? That brings me to calcium carbonate precipitation. So a little geochemistry. Wrong way. This is how calcium carbonate forms. You have water that has calcium in it, needs to have higher pH, and you'll precipitate calcium carbonate. Simple process. But what's really cool is when phosphate is around, that calcium carbonate will suck it up. It'll remove it from the water column. And in some lakes, that removal can decrease 80% of the phosphorus in the water column. So phosphorus is that nutrient that would otherwise fertilize the growth of algae and cyanobacteria. This can be a really great natural cleansing mechanism in lakes, streams, or rivers to remove that phosphorus. And indeed, folks have tried to take advantage of this as well by adding calcium carbonate to lakes and things like that to lower phosphorus concentrations with varied success. So in a lake, that lake has some calcium in it. The more photosynthesis that happens. So it's basically Le Chatelier's principle. You take CO2 out of the water column, use it for photosynthesis, you're going to drive the formation of calcium carbonate just through chemistry. Interesting biology happens in there too that I won't get into. But for now, this is sort of what happens. We call this a whiting event because it turns the water sort of a milky turquoise color. And eventually, right, that calcium carbonate or calcite with phosphorus glommed onto it will sink to the sediments. Now it's stuck down in the sediments and is usually fairly stable. So question is, does this process happen in streams? But before I get into it, I also wanted to say that calcium carbonate will form in a number of different ecosystems. Here we have a river system, there's another lake. It's actually been shown to form in sea ice and in wetland systems. There are a lot of great examples in Florida and in Central America where we see this calcium carbonate forming. The other thing you might be thinking of is that, I thought it was called something else. And you're right, there are a lot of names to describe calcium carbonate. This makes it really difficult as an ecologist to necessarily know who else has studied this topic, because sometimes it's called travertine, sometimes it's called tufa, calcareous, coral, reefs, microbialites, stromatolites. All of these processes involve the formation of calcium carbonate, but it goes by a different name. You might recognize some of these names and not others. Some of them are regional, some of them are due to the way the formation happens, things like that. But in terms of how I think about it, that absorption process can potentially happen in all of these, which is really kind of cool. Okay, so my work sort of sits at this geochemistry, biology, and the question that I'm going to dive into, right, is how does calcium carbonate deposition influence nutrient cycling in streams? And so I went to some streams in southern Arizona that precipitate calcium carbonate sort of like a cement layer on top of the rocks, which just sort of covers everything. This is similar a lot to the precipitation that we see here in Brazil too. So here to orient yourselves. So here is the US. The work that I'm going to talk about was done here in southern Arizona, and then up here in northern Mexico and Puerto Rico. Yeah. Okay. So this is another picture of the Arizona stream, which is to remind me to say that I'm usually talking to biologists, and there's a lot of plants and organisms that rely on these streams. It's an arid environment. So the streams are in the mountains, and they're important as a water source, food source for a lot of other organisms in that area. Okay, so here are study streams. And if one were to just go out and look at the chemistry of each of these streams, they would notice that there's a very interesting difference in the amount of phosphate in the waters of the three different streams. Phosphorus levels are low overall, but there is this nice decline. What you can't see is that this garden and Huachuca tend to have a lot more calcium carbonate depositing than this stream. Otherwise, these streams have the same geomorphology, same riparian zone, very other similar characteristics. Sort of begged this question was, is calcium carbonate actually responsible for that variation in phosphorus chemistry that we see? Matter. So in lakes, when we get the formation of calcium carbonate, that calcium carbonate falls to the bottom of the lake. Presumably we're no longer in the photic zone. There, so algae wouldn't be able to get access to it anymore. However, in streams, carbonate is still very much within the photic zone. So it does present this alternative hypothesis that could be happening, which is maybe calcium carbonate is absorbing that phosphate, but maybe it's just making it more readily available to the organisms that live right there on that calcium carbonate. Leaves open that question of whether or not this absorption process matters to reducing phosphorus availability. That's what I went out to test and to see. Okay, so here we are. We sampled a bunch of things, and through a lot of sampling, we found consistently that as calcium carbonate deposition increased, phosphorus concentrations went down, nitrogen concentrations actually tended to increase a little bit, and overall N/P ratios tended to increase. Carbonate deposition could actually be limiting biological growth because it also might be limiting nitrogen uptake. But again, this is all correlative. This is just what we see in terms of the relationships between calcium carbonate and these various elements. And the data is published in biogeochemistry. But we also went out and did some bioassays. And so we did some growth experiments, diffusing substrates, which is basically a really clever way to see what limits growth. You put nitrogen in one cup, phosphorus in another cup, nitrogen and phosphorus together in a third, and in a fourth, you don't put anything. And you look at the relative growth of algae on those different discs. And we found consistently that phosphorus limitation was related to calcium carbonate deposition. In these streams, calcium carbonate deposition is limiting phosphorus availability. However, the problem with this experiment, right, is that we're not actually looking at what's growing on the substrate. We're just looking at things in the environment. So we got to do more work. Okay, so we set out an experimental test and decided to go with the shade manipulation. So as I mentioned before, calcium carbonate deposition, right, is promoted by photosynthetic uptake. So if we reduce photosynthesis, we should be able to reduce calcium carbonate deposition, and then we can see what happens to the growth of the algae. Is phosphorus liberated and more algae respond to it, suggesting that calcium carbonate might otherwise be sequestering that phosphorus? Or is there no impact, right? Because maybe the algae can get at the calcium carbonate anyway, or can get at the phosphorus anyway that's in the calcium carbonate. So we can start to tease apart those two different processes. So our prediction is, right, if we lower calcium carbonate deposition, phosphorus availability will increase, potentially nitrogen availability will decrease, right? If we get more growth, we should see more nitrogen uptake. So we set up a shade experiment, and what I didn't include here, but is that we had for advantage of the fact that one stream didn't have any calcium carbonate deposition, and this stream did. So we put the shade up in both of those streams. So then we can use the comparison between those two streams, both having shade, to see what is causing shading a stream versus shading a stream with calcium carbonate. And the other thing we did is turning off photosynthesis so I can look at the algae response, right? This doesn't work. So under the shade, but what's happening downstream as well. So I use that downstream section to really get at, okay, if more phosphorus is coming available, we should see an increased growth in that downstream section. You're doing just fine getting that phosphorus, they're not going to respond to that increased phosphorus availability. So I'm just going to sort of walk through the results here. So we shaded the stream. We led to a 50% reduction in calcium carbonate deposition rates. So we didn't completely knock it out, but we reduced it in half, which was pretty great. That lowered phosphorus uptake rates. And with those lowered phosphorus uptake rates, we actually saw a bit of an increase in nitrogen. And again, because we're making this comparison between with calcium carbonate and without, we know that this reduction in calcium carbonate means that it was sequestering that phosphorus, because now we're not seeing that phosphorus decrease. We change in concentrations of either nutrient in the water column. This makes a lot of sense to me, just based on the scale of our change and the dilution basically that happens in the stream itself. But when we look at the things growing in that stream, we saw an alleviation of phosphorus limitation. And we saw an increase of percent phosphorus and percent nitrogen in the periphyte. The calcium carbonate is absorbing phosphorus in these systems, and it's reducing the access to the algae, despite the fact that they're so close together. So that was a pretty exciting finding. So we can kind of put this all together to look at what's happening in these streams, right? There's some source of nitrogen and phosphorus in the streams. And this is if there's no calcium carbonate around, right? You're going to get downstream flow, interacting with the sediments. Biology will take up those nutrients either way. Through algal growth, you get primary production and decomposition happening, and it can feed on up into the food web. All right, so then when we have calcium carbonate forming, we can adjust our understanding of the nutrient cycling in these systems, right? We're going to get sequestration of phosphorus in the calcium carbonates, which will reduce downstream flow. It's getting trapped in those sediments. Biological uptake. Biological uptake of nitrogen. So we're getting a bit more nitrogen flowing downstream than we otherwise would. And it sort of leaves this question of what's right. Their food source might not have as much phosphorus as maybe they need, or maybe it's just fine because maybe they're adapted to those conditions. But it does sort of ask this question of what's happening to the things that live in these carbonate systems. And we didn't answer that question in a direct way because I want to talk about another aspect of calcium carbonate deposition that I think is kind of fun. So calcium carbonate, it's an incrustation, right? It's not just interesting because it co-precipitates phosphorus. It's also kind of interesting because it blankets the benthos, right? And it's going to blanket anything else that's in the water. So here we have leaves, a leaf without deposition and a leaf with a lot of calcium carbonate deposition. And there's fairly good evidence out there that calcium carbonate reduces decomposition in streams because of the basically physical covering of those leaves. But the other thing it will impress are the organisms that live in the streams. And I'm also going to say that I am a horrible taxonomist, but I'm going to do my best. Okay, so here we have our wonderful water bug, and it's got a nice covering of calcium carbonate. And when my colleagues and I were working in the streams, we kept noticing that some bugs had calcium carbonate deposited on them and some didn't. And we decided to look at this question a bit more, and we discovered that back in the 1920s, Van Keer described some mothflies having calcium carbonate deposits on them and suggested that the process may not just be accidental. There might be bugs out there that are promoting the growth of these incrustations versus not. But it wasn't really well known, and there overall wasn't a lot of work done on this question. So we decided to dig into it. And here are some pictures of the different organisms. I already showed a water bug, but we have some soldier flies, water scorpions, and dragonfly larvae, all with some nice calcium carbonate crust on them. So Eric and I were doing a lot of the field work, and we knew Mike Bogan also had done a lot of collections of macroinvertebrates in these streams. We basically emailed Mike and we're like, hey, could you look through your collections and see which bugs have calcium carbonate deposits on them and which ones don't? And so his collections were from the same streams as well. And our hypothesis was, right, the longer an aquatic insect lives in a stream, the more likely that it will have calcium carbonate depositing on it if there was no selection of that growth. It should basically be a statistical problem, a chemistry problem. The longer you're around the stuff, the more likely you are to get it on you. And it turns out we were correct. So here is a big lunch of the organisms that we found that had calcium carbonate deposition on them and those without. The red arrows meant that we were the first describing it in a published article. And so that was pretty cool. It seemed to be related to how long these organisms are living in this stream. May not be selective. It leads to kind of another thing, right? So if you spend a lot of time in the water and you don't molt, in an important aspect of the biology there, you're more likely to have calcium carbonate deposits on you. This is problematic, right? Because if you're a visual predator and you get a big clump of calcium carbonate growing on your eye, presumably that's going to have a negative impact on your predation skills. Done by Ruth and Meyer showed that some of the macroinvertebrates that had calcium carbonate on them, others were less likely to eat them because they had essentially extra armor from that calcium carbonate. So again, depending on the life history strategies of these organisms, it could be problems or it could be beneficial. A lot done on that question, but kind of an interesting reminder that calcium carbonate might not just have interesting higher trophic level effects due to phosphorus limitation of food webs, but also because of these very direct impacts of the deposition itself. So down in mosquitoes, carbonate forms in these spheroid shapes. They're called oncoids, oncolites, microbiolites. And it's a biologically driven process. So there's this microbial community growing, and as it grows, it forms these layers after layers of calcium carbonate. From a scientist perspective, this is really great because here is an ecosystem I can pick up in my hand and put in a bucket and do some experiments on. So it's a very tractable way to study the impacts of calcium carbonate on microbial processes. So in this work, I was interested again on whether or not these microbial communities were limited by phosphorus, but I was also interested in whether or not there could be some organic carbon limitation as well to respiration to the bacteria. Thinking about organic carbon, because these are really oligotrophic, pristine systems. So they're likely candidates to be organic carbon limited as well. Okay, so looking at phosphorus limitation of growth, I added phosphorus and saw an increase in gross primary production, suggesting again, calcium carbonate might be is that I saw responsive respiration as well. So bacteria were also and to an extent of carbon as well. So are microbes associated with the calcium carbonate phosphorus limited? Yes. And then I went ahead and repeated that same nutrient diffusing substrate experiment here, where I was just trapping the sort of free living microbes to see if those microbes were phosphorus limited. So here's a picture of those, that experimental design. And I found out that no, the microbes in the system that weren't directly associated with the calcium carbonate were not phosphorus limited. So suggesting again, the power of calcium carbonate in driving phosphorus dynamics in the system. Again, these are correlations, not direct manipulative experiments. So I did some work to manipulate calcium carbonate deposition in this oncoids. And yeah, so I took advantage of the fact that I could work in buckets instead of stream reaches to be a little bit fancier in how I reduced calcium carbonate deposition. So I repeated the decreased photosynthesis way that I had done in the streams, but then I also just removed calcium from the water. And I did that by running the water through a water softener, replacing those calcium ions with sodium and potassium, and then incubated them for 6 weeks, basically saw what happened. This would have been a really nice experiment to run in the streams in Arizona, but it's entirely impractical at a reach scale. So when you only have to soften 20 liters of water, it's a lot easier than thousands. Okay, so what did we find? For organic matter accumulation, as total biomass on these systems, we have our control here in the middle, and calcium reduction. You see a slight increase of organic carbon when we remove calcium, and a complete knockout of organic carbon when we turn the lights off, but some growth and recovery after the first couple of weeks. For phosphorus, we saw a really dramatic increase in the amount of phosphorus when we stopped calcium carbonate precipitation and left the lights on, versus in the dark, it pretty much just stayed. It was clear that the photoautotrophs in these systems could not handle in the dark, which makes sense. And that while we did get some bacterial growth, like we just completely changed the microbial community here. But with for the increased phosphorus availability, more sort of proof that this mechanism, calcium carbonate, co-precipitation of phosphorus, reducing bioavailability to organisms in the system, is happening in a completely different system. So just to sort of summarize, we found that microbes respond quickly to changes in phosphorus availability. Photosynthesizing microbes are really important in those systems, but there is some things happening with the non-photo autotrophic organisms in that system. And the calcium carbonate, right, deposition is lowering phosphorus availability to the microbes. All right, so in summary, did a bunch of work about what was going on, lowering phosphorus flows downstream in the Arizona streams, had some implications for nitrogen cycling, potential and actual phosphorus limitation of biological growth, as well as the constraints on biological uptake of phosphorus in the Rhea mesquitase system. So going back to my bigger question, right? Can we make calcium carbonate deposition happen in every ecosystem and be an effective sink for phosphorus? Well, yes, kind of, in at least these systems that seem to work well. Is it a permanent sink for phosphorus? My experiments were short term, so I can't really answer that question yet. But it does suggest that if there is an ecosystem that's depositing calcium carbonate, we should be managing to keep that calcium carbonate happening. And what does that mean? So something I didn't mention before is that calcium carbonate, if there is too much phosphorus in the water, that's actually going to stop that geochemistry from happening. Calcium carbonate will not form because too much gets absorbed on the ions, and so you lose that negative feedback on phosphorus concentration. So we can go to Lake Mendota to kind of look at that question. So Lake Mendota is one of the longest studied lakes in Northern America, in the US. And the lake used to have a lot of calcium carbonate deposition happening based on what we can see in lakeside. A lot of whiting events. But after the watershed, after Europeans sort of moved in and started farming, cutting down a lot of the forests, the lake entered a very different phase and stopped having dramatic calcium carbonate deposition events happening. Starting to see some re-emergence of calcium carbonate. So, and here's sort of a blown in of the recent re-emergence of those whiting bands. That the deposition stopped here is not due to a loss of calcium in the water. As far as we can tell, calcium concentrations in the water remain consistent, but phosphorus concentrations increased. And so we think that this is a reflection of the loss of calcium carbonate because too much phosphorus got in the system. However, started to get really concerned about water quality in Lake Mendota and started implementing a lot of management strategies that started to reduce phosphorus concentrations. We think those phosphorus concentrations went down just enough to allow calcium carbonate to start forming again. And what's great here is that we have this lake sediment record paired with lake sampling record. And so we know when the blooms happened and we can match that up to when we see it in the sediment core as well, which is kind of cool. Okay, so in more recent times, index, it's basically a proxy of whether or not calcium carbonate should be forming. We saw between 1995 and 2000 a nice decrease of phosphate concentrations. Marked a year of a really profound whiting event that seems to have almost permanently knocked down phosphate concentrations in the lake. So we're pretty sure that whiting events are a good job of reducing phosphorus in Lake Mendota. Right, if those phosphorus management strategies stop and phosphorus starts to come into the lake again, we suspect that the lake is going to go back to this system where calcium carbonate won't be forming and phosphorus concentrations will increase just that much more. Okay, and I also wanted to make one note on the other caveat to this story. So I basically just said calcium carbonate absorbs phosphorus. I didn't talk about the rate at which it does that or the percent at which it does it. So if you have, you know, X tons of calcium, you should expect X tons of phosphorus in there. Did look at that question, and they have a paper published from the 90s that basically has this relationship. The concentration of phosphorus on that ratio of phosphorus to calcium carbonate precipitation. Their model tends to grossly overestimate the efficiency of calcium carbonate absorption. So this is actual lakes that Jake and I consolidated, and we see that there's a fairly consistent lower rate of removal of phosphorus from calcium carbonate. That's all I remind you to say. I think this feedback loop is super, super cool, but there's tons of caveats in how it works. But, oh no, okay, so sometimes when things go from map to PC, we lose data points. Here's a map of the world. It used to have some points that were studies of calcium carbonate to show that it wasn't just a handful of places in North America that I talked about in here, but calcium carbonate deposition happens all over the world, generally in any aquatic system that's fed by a carbonate aquifer, so limestone aquifer. We tend to see calcium carbonate forming in the surface waters. But you'll just have to wait until I publish that at some point. Okay, anyway, so I'm really excited to be here to look at some of the travertine streams in the area. And that's it for my talk, and I am happy to take any questions.
Thank you so much, Jess, for coming.
Really good question. Yes. So in the Arizona streams, let's go back to the Arizona streams do not have any fish, which is part of the reason that it's really easy to look at the macroinvertebrates, because they're just way more macroinvertebrates than you would ever see when fish are around. There's, a nice host of macroinvertebrates in those streams. And in Rio Mesquites, there are fish. Cichlids are probably the most common. There's an endemic cichlid species in there. There have been some introduced bass and some other fish. There are snails in both systems. Snails, I see more of them in Cuatro Ciénegas. And it's actually some interesting work on the snails, because the snails graze on the microbialites. And one of the early hypotheses of why we don't see more microbialites. So microbialites or stromatolites used to exist all over the globe. But then evolution happened, herbivores came into being, and the idea is that herbivores would chomp on microbial communities that form these at a faster rate that they can grow. So that's why we don't see them anymore. It's not entirely true, as all things in biology have exceptions to them, because here we have microbialites that snails do chomp on them at about I think it's like 80 to 90% of their growth gets consumed by the snails, but they grow at a slow rate, even despite snail herbivory. There are also microbialites in Germany that have snails on them too, and they grow just fine. So there are certain conditions that it seems to be okay. I haven't seen mussels in either system. Yeah, so my PhD advisor, Jim Elser, has done a lot on that question. And he was really interested about these snails because the algae on here tends to have a carbon to phosphorus ratio of about 500 to 1. Generally, algae grow at more of like a 100 to 1. So, 5 times as fewer phosphorus atoms are encountered by the snails that live on these oncoids. So how do they survive? So they did a bunch of experiments where they added a bit of phosphorus to the microbialites to increase that percentage of phosphorus, and then saw snail growth. And to an extent, snails do better when they get more phosphorus, but then if they get too much phosphorus, these snails, their growth declines, they were dying. And so they saw this, it's sort of a classic physiological curve. They want it to be at a certain ratio. If they're above that or below that, they don't do well and they die.
For the Corman said now. Very low carbon production.
The brown pathway is also really important in these systems. They get a ton of leaves. And so you do see a lot of shredders and things too.
So they-
Yeah, at least in the Arizona. Yes. The fish in Rhamescetes, they're cichlids, so of course they have like 1,000 phenotypes. I think it's only 3. One of them is effective at eating the snails, but another one is tends to eat the microbialites. And my colleague Steve Thomas has seen fish munch on travertine in Trinidad. So we think some fish might have interesting physiology going on, where maybe they're getting nutrients from the calcium carbonate. Maybe they're getting at that phosphorus that's stuck in there. But it's something we've been thinking about. Yeah. That's the open question. Yeah. So the fish in travertine,
Maybe it can.
and to an extent the fish in Rhamescetes too, like they poop and it's just all white, right? So the calcium carbonate is going through their system. Whether or not they're taking the phosphorus from it is an open question.
So I've seen in Trinidad in the farm. So we had like actually armor catfish, zebras, and you could see their poop all around white.
Right,
So
right.
that would be trying to collect invertebrates in the travertine. Diversity of invertebrates. So it was difficult to find them, and it was when I was at the time I was thinking, well, maybe it's because the microhabitat is so different.
Yeah.
So then maybe like the community does change. Rocks,
Yep.
leaves, but you have mostly all of these travertines, mostly of the all of this cement added on top of the all of the substrate.
Yeah.
So maybe it's more difficult for some species to be able to. Because exactly, like maybe they don't have enough food.
Yeah, I think certainly the lack of habitat is important. I would guess it's probably potentially a primary, depending on how the calcium carbonate forms, because sometimes it's sort of patchy within it, and so you can still have, it's not a complete cement layer, it's sometimes just on some. But I think it's a good question too, like what are the food web implications? What are those top up or bottom up versus top down pressures? Yeah.
I think sure.
Yeah, yeah.
Like you pointed out that substrate there, you really measuring the amount of
Yeah, yeah. And that's part of the reason that I found this question so exciting, because it was like, there's a lot of the work in wetlands in the Everglades and down in, oh gosh, I'm not going to remember. Anyway, but Alicia Rejman-Kova's work in Central America, where the calcium carbonate forms during the day and dissolves at night. And so when that dissolution happens, they see an increase of phosphate at night and an uptake by the microbes. So there's that cool pattern of, you know, the microbes are benefiting from that change in pH with photosynthesis and respiration. But then in the microbialites here, one of the geomicrobiologists on the project, around Garcia Pichel, like saw the cyanobacteria actually boring into the calcium carbonate. And so there's that active dissolution where, yeah, algae and cyanobacteria, they're really good at getting at things, which is why looking at the ecosystem scale, I didn't know that I'd necessarily see that response to phosphorus. I was like, oh, well, they should be doing just fine. And they might be, but they're still limited. Yeah. There's a lot to be done. And if we only had stable phosphorus isotopes, we could really get at that, what's happening. Yeah. What?
radioactive phosphorus.
No. So I looked into using O18 labeled phosphate and doing some nanosims mapping. But it turns out that O18 gets, like, if there's any alkaline phosphatase in the system, it just gets exchanged and you just dilute the signal with water. So it doesn't work very well. Yeah, I'm potentially interested in doing some P32 work. Radioactive work is always, yeah. Radioactive. Yeah, so in marine systems, the mechanism at which like coral reefs are formed is different than how it's forming here. And so you don't see that phosphate absorption happening in the same way. Directed process. But just like we see in the oceans, increasing acidity, decreasing calcium carbonate deposition could happen in these systems too. The thing about inland waters is in a day, they tend to see such drastic pH ranges that the changes of atmospheric, like they'll be super saturated with CO2 and then undersaturated. And so that atmospheric signal doesn't tend to be as strong. The caveat is there's only been a couple actual studies on it. My guess is that in these systems where it's biologically driven, it's probably going to be a bit more robust. We might see it in a slightly different place. But I think it will be more robust. But again, it also depends on how far we push atmospheric chemistry. At that point, it might be something else. But yeah, no, it's a good question. The work that I've seen is in the Great Lakes, in the Laurentian Great Lakes, where people have looked at the models of the summertime precipitation events and how yeah, so there's been a fair amount of work in Canada. They have eutrophication problems and also a lot of carbonates around, and scientists and some work in Europe on this too. And the three basic ways people have gone about it is one, just dumping calcium carbonate into the lake and hoping it sucks up that phosphorus. Or the two main ways doing that, or changing the pH of the lake to promote calcium carbonate deposition, so trying to increase the pH. When you just dump the calcium carbonate, it will suck up some phosphorus, but it doesn't seem to be as efficient. It kind of will trap phosphorus in the sediments a little bit more, but you don't get the loss in the water column. It seems to be more efficient when you promote deposition or promote precipitation in the water column. So if you can increase the pH or add something to cause the phytoplankton to grow a bit more, to cause calcium carbonate, you do a better job of sucking up that phosphate from the water column. From what I've read, you usually need to do that to a lake like for several years in a row before you see the benefits. I haven't seen follow-up studies like 5-10 years down the line what it works, but there is it seems like there is some benefit, some possibilities of it in certain lake systems, which is kind of exciting because it's like you're promoting a natural process, so you're not adding something artificial to the system. And yeah.
Sam, it's an impact, like the positions.
Yeah, so that's a good question. In lakes, I don't know if anyone, so an issue in lakes because that'll be in the pelagic zone where most of it's happening. But I mean, I think that's a good question. And what are the system effects? Yeah. And I haven't seen anyone try it in a river system, like, or a stream. Yeah. Sure.
Hi guys, thank you for
Yeah.
So I was wondering about the fossilization process.
don't know. I don't think we have that data. So on the insects themselves, if there's more of it now, is that what you're asking?
Yeah.
All right, okay. Yeah. Yeah, I mean, that seems reasonable. I don't have any data on it or observations. Yeah. Yeah. Yeah, and let me know.
Of this large predator fish offsprings, some tools that are from. So I love what was supporting it, and either it has from how much of the allotments matter.
No, that is a great question, and something that we've chatted a little bit about. And I mean, some interesting things, right? Our aquatic systems, it's not uncommon to see those inverted pyramids. There's one thing. The other thing is that in carbonate systems, there might be something really unique about the fact that your phosphorus is retentive in the system because it's stuck in that carbonate. So as Tim mentioned, microbes are really good at getting at things. And so it could be that over time, that leads to this very high phosphorus use efficiency, leading to, you know, not necessarily high biomass rates, but sustained growth that then can trickle up the food web. But that is all hypothetical. But it's something that is not uncommon in carbonate systems to have these higher biomass. Possibly, yeah. Yeah, Kara really like to grow in carbonate systems and
Plus the population and start.
right. Yeah. I think they're super exciting systems. There's so much we don't know. And to your point, they're often culturally or culturally important ecotourist sites, things like that. So yeah, they're important to understand.
Well, the next step to that, of course, was, because we're very approach in the lab, wouldn't it be great to do production was very common, very
Right.
it's really
Right. And two, I wonder, thinking more about scale of these experiments, and maybe that is really important too, and seeing the mechanisms and the feedbacks actually play out as expected.
Yeah, so these are strict. They have a lot of phosphorus input.
Right.
Relatively.
Moderate. Yeah.
And so then you get to one point, it's like you get to a threshold basically. And then you get like when you go over a certain threshold of phosphorus, buy too much phosphorus, that prevents the position. That's in carbonate.
From what I understand it, it's as that there's actually like a couple stages involved in the mineral formation process, but calcium carbonate can handle a certain number of kinks in its crystalline structure because of that phosphorus bonding on. But if too much phosphorus gets on, it can't, the crystalline structure just doesn't form anymore. So you don't have that stability.
Of phosphorus concentration is also system dependent.
Yeah, so a scientist, William House from the UK, has numbers that he's set that are there, but based on like that lit survey and things that I've done, I would tend not to hold too tightly to those numbers. But it does seem once you get into that eutrophic zone, then you're in trouble. Yeah. The other thing that happens, and so what I just described is sort of the chemical mechanism, but one of the things that verify in calcium carbonate wetland systems in Florida, they see the loss of calcium carbonate, they attribute it to a turnover of species composition. So you're getting different types of algae and algae that no longer promote that calcium carbonate deposition in the same way that other species did. Yep. So it all goes back to this equation. So when the equation is going in this direction, and there's because in chemistry equations like to be balanced, if you remove a compound or a molecule from one side of the equation, the balance will tend to move into that direction. So with photosynthesis takes up carbon dioxide and turns it to sugar. So it's taking up the CO2. So that means that this calcium carbonate wants to bind with this bicarbonate to form more CO2 to balance out the loss. So that's what's driving the equation this way. So there's that connection between the biology and the chemistry. That makes sense?
Yeah, it's like respiration.
Exactly, because respiration pumps a bunch of CO2 into the system and then it is gonna move it. Yep.
That's why you can. Process changing between night and day.
Exactly. Yep. Yep. All things of biology, it's not a perfect one-to-one relationship, but that's the general trend that you see. Yeah. And it's in the opposite way that you would expect.
Oh,
The calcium carbonate likes to form at high temperatures.
high temperature.
Does not like to form at cold temperatures.
That's why there's more in like thermal, like volcanic stream.
Então, sobre a biologia, então, em relação com a biologia é mais na prática mesmo. Então, seria mais uma dúvida da metodologia usar a relação ambiental que teve com o rio que botar. Eu queria saber se teve alguns outros dados fora para saber tipo, temperatura, entrada de para saber se essa cobertura gerou algum outro impacto que afetasse o resultado dela, e se ela teria alguma outra adaptação para pesquisa, se ela fosse fazer de novo ou em outro lugar. Igual.
Light, so the light was the mechanism that called. Want to know if you think that viable that could have affected your.
Yeah, that's a good question. I read a bunch about shading experiments before I did this one. And so one of the things you'll notice is that the shade is a little bit above the stream. That's to make sure that any leaves or stuff coming in from the watershed could still get in there, because we didn't want to block leaf input because leaves are an important part of the stream. We also monitored temperature within the stream and saw very little effect of the stream, of the shading on stream temperature. So we were pretty confident we weren't manipulating that variable. There were 30-meter stretches. So it wasn't a huge stretch, but it is possible we had unintended effects. But I was most concerned about temperature and like leaf inputs, and I think we handled those two okay. Yeah.
And I was wondering if this incrustation that big impact.
Yeah. So here's this giant water bug with eggs on its back with calcium carbonate on it. It's a great question, and I have no idea. Yeah, it's a great question. It certainly I mean, yeah, it happens, whether or not it matters. Yeah, it's a good question.
And hope is managed.
Those are great hypotheses. I have no idea. Let's test it.
Yeah.
Well, the harmony.
Right. Yeah, this one that has it on its eye, we sort of were wondering if it like was sick or something. Because I don't know. It was very unexpected. But yeah, I think the color change is
Those are thoughts.
yeah, I mean, she seems to find a way always. And if there's an empty niche, it'll fill it. Right.
Very much. It was super cool and also interesting questions. So well,
Não, só Maria Alice que agradeceu o